Let's discuss structure of nucleosides and nucleotides. When we talk about the control hydrolysis of nucleic acid, we get nucleotides and subsequently nucleosides. So nucleosides on hydrolysis with inorganic acid yields sugar and base. They are glycosides formed by combination of ribose or deoxyribose with purine or pyrimidine bases. Nucleosides are non-reducing beta glycosides. That suggests the anomeric OH is not free. It means the C1 dash of sugar is bonded to the nitrogen atom of the base. The C1 dash carbon of sugar residue is attached to nitrogen at 9 position in the purines, whereas it is attached to nitrogen at first position in pyrimidine bases. Now to understand it better, just look at the structures of sugar. These are the two sugar forms, 2-deoxy-D-ribose, which is found in DNA, and another is D-ribose, which is occur in RNA. The numbering in sugar is done in this manner. The carbons are numbered like 1 dash, 2 dash, 3 dash, 4 dash, 5 dash. Whereas the structures of bases, when we talk about the two different class of bases present in nucleic acid, purines and pyrimidines, the numbering is given 1, 2, 3, 4, 5, 6, 7, 8, 9 in purine, whereas 1 to 6 in pyrimidine. There are two types of purine bases, adenine and guanine, and three different types of pyrimidine bases, cytosine, thymine, uracil. Thymine is occurred in DNA, whereas uracil is found in RNA structure. Now talking about the structure of nucleoside, just take an example. The D-ribose sugar and adenine. Now we have discussed that the sugar attached to nitrogen at the 9 position in the purine bases. So for the adenine, the nitrogen at this 9 position will get linked to the C1 dash carbon. It can be represented with this, and the nucleoside form is known as adenosine. Here, this bond formed between the carbon and nitrogen number 9 in the purine is also called as beta N glycosidic bond. This adenosine is present in RNA. Okay. Now let's discuss for DNA. For DNA, the nomenclature is 2-deoxyadenosine. Okay. From the structure, we can understand at the 2 dash position there is no hydroxy group present. So this can be formed when we have 2-deoxy-D-ribose and adenine linked together through a beta glycosidic linkage. So DNA will have 2-deoxyadenosine as one of the nucleoside unit. Now when we talk about the combination of sugar with pyrimidine bases, okay, we know that in RNA we have uracil. So D-ribose sugar, when form a glycosidic linkage with uracil, it gives uridine. This particular nucleoside is present only in RNA, as uracil is the pyrimidine base found in RNA only. Whereas when we form a nucleoside from 2-deoxy-D-ribose and thymine, it gives thymidine, that is found in only DNA or deoxynucleic acid. So this 2-deoxythymidine or thymidine is result from combination of 2-deoxy-D-ribose sugar and thymine in DNA structure. For the remaining structures of nucleosides, okay, in RNA, we'll have guanosine and cytidine, whereas in the DNA, we have 2-deoxyguanosine and 2-deoxycytidine. These are the respective nucleosides present in DNA and RNA. Now let's discuss nucleotides. Nucleotides on hydrolysis with aqueous ammonia at 175 degrees Celsius yields a nucleoside and phosphoric acid. Therefore, they are also called as phosphoric esters of nucleoside. The phosphoric acid is directly attached to sugar. In monophosphate, the phosphoric acid attached to carbon number 3 or C3 dash or carbon number 5 or C5 dash position of sugar residue. In other words, we can say that the phosphoric acid form the ester linkage with the hydroxy group at C3 dash or with the hydroxyl group present at the C5 dash in the ribose sugar. Therefore, the structure of nucleotide can be represented when we have, for example, adenosine, a nucleoside combined with phosphoric acid. Now, if it attach to the hydroxy group at 3 dash position in the sugar, we get adenosine 3 dash phosphate. Or if it attach to the hydroxyl group present at the 5 dash carbon in the sugar, we get adenosine 5 dash phosphate. So these are the phosphate esters formed when phosphoric acid reacts with hydroxyl group in the sugar at 3 dash position or at 5 dash position.